Thank you everyone for joining us. Without further ado, it is my pleasure to introduce our speakers. Adrian Ramirez Galilea is Associate Director of Automation and HTE at Takeda Pharmaceuticals in Cambridge, Massachusetts. Adrian brings a wealth of experience from work at Swisscat, ETH Zurich, and King Abdullah University of Science and Technology. Adrian holds a PhD in chemical engineering from the University of Zaragoza, Spain, and his work has been widely published and cited with significant contributions to the fields of catalysis and chemical engineering. Dr. Germán Tribucait is the co-founder and CEO of Atinary Technologies, and he has led significant R&D initiatives and authored influential reports on innovation and policies related to technology and the environment. Germán has a BA in economics from ITAM and a PhD in economics and finance from Harvard. In his latest venture, a deep tech startup, Germán is looking to transform R&D and molecules discovery. And last but not least, Alex Waked earned his PhD at the University of Toronto, studying main group catalysts for organic transformations. As the lead for Catalyst D2D software, Alex supports projects involving automation of data, high-throughput experimentation, and the implementation of new technologies for the digitalization of chemical and analytical data, its management and preparation for machine learning and AI. And with that, I am going to hand over to Germán and ask him to take it away from here.
Thank you, Sanji. So can I share my slides, please? There you go. Can I share?
Is it coming up? Okay, let me.
There you go. Thank you. Excellent. Thank you, Sanji. Hello, everyone. Thank you for joining us for an exciting session on the future of R&D and the rise of self-driving labs. I'm Germán Tribucait from Atinary Technologies, where we are reimagining scientific discovery. New technologies are reshaping how we work. In the case of R&D, manual, time-intensive experimentation is being augmented by automated, smart, and iterative workflows powered by AI and robotics. Let me tell you a little bit about Atinary. We built a platform that makes AI-driven R&D a reality today. With SDLabs, our no-code AI platform, scientists can explore more with less experiments. That means you can screen much larger chemical spaces and solve more complex optimization challenges with multiple parameters and multiple objectives. Whether you're working with categorical variables and want to add physicochemical descriptors or constraints, SDLabs adapts to your needs. We use machine learning to help you find the optimal results with a fraction of the experiments and a fraction of time and cost compared to the traditional trial and error methods like design of experiments and one factor at a time. You don't need to worry about stability, maintenance, and scalability of your algorithms. And we also offer state-of-the-art cybersecurity and data and IP protection. No coding and no AI experience is required to start optimizing your experiments with machine learning in your existing workflows today, with or without robots. Moreover, you don't need to wait to finish cleaning up and curating your existing data sets. You can start from scratch with no data and start building your digital, reliable, and reproducible data sets along the way. Atinary partnered with ACDLabs to make it even easier for R&D teams to leverage digital data sets, train AI models, and integrate all lab equipment. As you will learn today, combining Atinary SDLabs with Catalyst and robotic platforms produces high quality and optimal results much faster than ever before. Alex, welcome. Please tell us more about Catalyst and the exciting work that ACDLabs is doing in this space.
Thank you, Arman. So in recent years, there's been a very strong push towards the digitalization of chemical studies. So this is vital for the automation of HT processes in the lab, and allows for integrations between hardware and software applications. It's also important for data science applications as well, like for developing machine learning and AI models, another very hot topic in the HTE world. So the ACDLabs Spectrus platform is both software and hardware agnostic, right? So which allows for easy processing and interpretation of analytical data that's collected from almost any vendor's software. So as you can see on the top left-hand side of the screen, there's a list of different vendors that we work with. So the data is digitally stored, allowing it to be searchable and used in integrations with third-party systems. So ACDLabs has been doing analytical data handling for more than 20 years now, so a very long time. And in the last few years, we've started to begin working on developing integrations with chemistry hardware as well. So some examples you see again on the top left-hand side of the screen there. You'll see some examples like easy mass reactors, Juniors, Quantos robots, and more there. So the Catalyst D2D application is built on the Spectrus platform and brings all of these features essentially to the HTE lab. So the software takes the chemist through the entire HTE process, starting from the experiment design all the way through to data analysis and interpretation. So Catalyst also integrates with third-party software applications, including robots, analytical instruments, and more relevant to today's topic, the Atinary SDLabs platform. So the Atinary Catalyst integration can be split into 3 general steps. So first, the SDLabs assists the chemist with the initial experiment design. Catalyst then supports experiment execution and data analysis. And finally, the data is consumed back into the SDLabs platform for further experiment refinement using the machine learning models. So this process repeats in a complete feedback loop, as you see on the screen here, and it'll be demonstrated in the full capacity by Adrian very soon.
Thank you, Alex. Now it is my pleasure to introduce our keynote speaker, Adrian Ramirez Galilea. Adrian is at the forefront of applying AI and robotics in pharmaceutical R&D. At Takeda, he and his team have built next-generation workflows that go beyond manual experimentation, connecting digital data, machine learning, and robotics into fully integrated design, make, test loops. Today, Adrian will walk us through how this approach is enabling faster, smarter, and more cost-effective R&D. This is not just about accelerating innovation. It is about augmenting what's possible in chemical research and helping shape the lab of the future to tackle some of the biggest challenges that we face as humanity. Adrian, the floor is yours. Thank you very much.
Thanks a lot, Herman, for the nice introduction. So today we would like to show you the recent work we've been doing at Takeda, enabling self-driving labs for synthetic molecule process development. But before jumping on this self-driving lab topic, I would like to give you a bit of background, of who we are and what we do and why we are pursuing this self-driving lab approach. So first, a bit of history about Takeda. It was founded in 1781. So it's quite an old pharmaceutical company with the HQ in Tokyo, Japan. It's one of the top 20 pharmaceutical companies with around 50,000 employees. And the therapeutic areas include oncology, rare diseases, plasma, vaccines, neuroscience, and gastro. Inside Takeda as a whole, we are part of the synthetic molecule process development, or SMPD, which is a process chemistry and engineering group located within R&D. We have around 100 FTEs in 2 sites, so one in Cambridge, DMA, and one in Sonnen, TISO. And we only focus on synthetic molecules. So from all the prior pipeline, we will tackle the synthetic molecules one. which comprise small molecules, peptides, and oligos. And our work spans from early process development to process optimization and tech transfer. And we support this pipeline by finding new routes, so improving those routes, so they can be transferred for clinical trials or manufacturing. We scale up some existing routes to provide materials for clinical trials, and we also do tech transfer to our manufacturing partners. And inside SMPD, we have our high-throughput and automation team, so we are embedded into the process engineering group. It's a relatively new team. It's less than 3 years old. And prior to 2022, there was no dedicated high-throughput or automation group at SMPD, only some isolated SMEs and equipment, with most of the work being externalized. So all of that you are seeing today has been developed in a bit less than 2 years. So it can be done. Then our team, right, aims to provide support to all global SMPD, and our focus areas include the main workflows, right, of our team, which are solubility, crystallization, and liquid extraction. We obviously do a lot of reaction screening and optimization. And in addition, we do some custom automations and some robotics projects that you will later see. And this is our team. So it says myself, then we have Andrew Cukor, which is an automation scientist, Dimitrios, which is our robotics engineer, Uta-san, it's the Japanese side of our automation scientist, and then Philip Tran is our newest robotics engineer. If you've seen our labs our new labs in Cambridge, we have two differentiated areas. So we have the main high-throughput labs around the Unchained Labs family of tools. So we have here 4 juniors and 1 OSR. They are in a set of 2, so we have 1 powder junior and 1 liquid junior, and duplicated, and they are inside a custom-made isolator, as you can see here on the left. And then we also have a robotics development area where we carry out some custom integration projects ourselves. So in the image on the right, you can see a dual-arm Yami in a Pentium frame. And this lab is currently being replicated in our site in Sona. And one of the beauties of this workflow is that once you have everything done, even it has taken us 2 years to reach this state, now we can very easily replicate our labs in our other sites, because we have all the knowledge and the know-how. So, a bit more of background on our historical problem statement and why we are trying to do what we do is that in the past, process development, and I think it still is very labor intensive, right? You have to do a lot of trial and errors, a lot of screening and optimization efforts, and they were typically done in vials with the DOEs being experience driven by our scientists. And this resulted in SMPD being heavily outsourced because we didn't have the numbers to run all of this internally. Then if you outsource some of this work, often we have no clear understanding of the methodology employed by our partners. So how many samples they were evaluated, what certain variables were selected or discarded. You typically just receive a report with a few slides with the best performing candidates. And of course, the data is not fair, right? You don't have typically access to all the data, from the analytical data to the process data of the reactors. And then one of the issues that we have, and also this is, again, if we do internal, it's a bit minimized, but it also happens that after we complete a project, the data is not stored in an internal databases, right? The knowledge is lost, and typically we duplicate efforts across projects. So unless we have a scientist that joins a similar project where they have faced a similar issue, we typically have to start from scratch. And on top of that, we are a relatively lean organization with not so many contractors or FTEs to run internally all the required screens in the standard manual approach. So if we want to change and shift how this works, our solution was what we call the self-driving data-rich HTE that we are trying to apply here. And now we are going to dig a bit more on this workflow that I will explain point by point. So here you can see a scheme of how we envision this workflow, what we are trying to build. So let me put the pointer. So this is a closed loop, multi-generation, self-optimizing workflow, but the main difference, right, is that it's not just the closed loop. But we start, the way we envision it is that we start in our ELN, in this case mathematics, where a scientist introduces a reaction, in this case, for instance, that they want to optimize. And then that reaction, which is just the smile codes of your product, your starting materials, etc., right? That gets translated into a reaction database where we have all of our prior knowledge. And there is a sort of retrosynthesis tool, although it's more a parameter recommender tool, that looks at that reaction database, at all of our prior knowledge, and then it's able to tell us if we have done something similar in the past, and more importantly, what parameters should we vary in order to optimize these reactions. Because most of the time when we are doing reaction optimization, we tend to use the same bases, the same solvents, the same catalyst, and we can have the best closed loop with the robotics, with everything in the world. But if we constrain our parameter space and then we left out some of the catalysts, some of the bases, some of the solvents that do actually work, we might be discarding some pathways that do actually work, but we simply are not able to optimize them, because here in the first step, what we are doing, where we are setting the boundaries, right, our parameter space, we are missing some key elements. So this prior knowledge coupled with all this retrosynthesis is where we try to minimize that in order to have a proper data-driven DOE, not just experience intuition-driven DOE. Then once we have that, we need to translate that into an automated reaction screening, in our case with the ACDLabs family of tools. We are also working in the automated consumables handling, because this is also something that typically it's overlooked. And if you run this 24/7, you will start to generate a lot of waste that you don't want manually to take care of. So we are also working on this side. Then we have the data-rich analysis. So we call it data-rich HTE because it's not just we take one sample for HPLC. We can take HPLC, but we can also take Raman, we can take Vision, and we can also take XRD if needed, right? If you are doing some crystallization, that will be the same loop, right? So it's more than just HPLC. Then we need a way to automate the data collection and visualization. That's where Catalyst kicks in. And then we also need to send this data to our AI optimizer, right, to predict the next generation. What should we test next? So that's where we are using Atinary for. So then we can carry out this loop 3, 4, 5 times until we reach the maximum. And this is our standard business practice. So we don't do factorial DOEs anymore. We always go through this closed loop workflow, and it has been demonstrated to be really, really robust and efficient. But in order to be successful in this approach and have a paradigm shift, we need a smart combination of hardware and software. This is not just I'm gonna buy a few robotic platforms and alas, all my problems are solved. You need to also look at the data automation to remove all the bottlenecks, like analytical data process or software hardware connectivity, because here we are looking to end-to-end lab automation. So it's a smart combination of both. And most of the times the data automation it's overlooked because it's under the scenes, right? And it's not something that you see in the lab where you go to do a tour, right? You see all the robots, but not all the data connectivity that needs to be in the back for this approach to really be successful. And just to give you, one example of the integrations that for our workflow we need to have. So it's quite a lot of software layers between the physical and digital automation, with a master scheduler on the top. So we start with this retrosynthesis database that predicts the conditions. Those conditions needs to go to Catalyst to build the reactant tables. Then those tables goes to Atinary. Atinary produces the first generation of samples to be evaluated. Catalyst translates that into the ACDLabs robots. They perform the experiment. Then we have the mobile robot that moves the samples to the UPLC. The UPLC does the analysis and goes back to Catalyst for processing. Then Catalyst sends that information to Atinary to predict the second generation, then back to Catalyst, right, to create the new unchained file. And then we have the mobile robot here transferring new plates and removing all plates to the juniors. And on top of this, you need a master scheduler that's able to trigger all of this autonomously, right? In our current state, we are almost there regarding the integration. So you can see that only a few of them are being developed, and we are also developing the master scheduler. So it's a journey, and there is a lot of very small tasks that need to happen. So now we are going to dive a bit more on both sides, in the physical automation and in the digital automation. So for physical automation, the first approach we took, right, was to standardize the size and format of all of our screens, moving to bigger formats and lower throughput, 24 by 4 ml, instead of the standard HT practice of 96 watt plates. And we not only standardized the plate format, but also the positions in the robot deck, right? So we will always have the solvents in the same positions, the reaction plate in the same position, the caps in the same position, because this means that we will have less errors with the robots. And we can, by applying this standardization, it's much more easier for us to reach this autonomous self-driving loop. Also, in this lower throughput plate, we can gain confidence in the data, right? Because we scale up 10 times to reduce manipulation errors. We can overcome the loss of throughput, so we don't care anymore about big format plates, because we are using this AI guide optimization with the Atinary algorithms from one generation to the other. So this means that despite using a smaller format, we can more efficiently explore the parameter space. We can sample several times without disturbing the reaction, so typically we sample 4 times. Now we can fully automate the workflow, leveraging mobile robotics, as we will see, and then we can do a more data-rich despite being SD workflow. So we not only take HPLC, but also images, as you see here, and also we have non-contact Raman we can take in the chain by moving the vial to the Raman. So we have a lot of information for each data point, not just a hit. Then, this standardization is what has allowed us to automate the workflow with the ACDLabs robotic platforms. So this is what I described, right? So we start with the powder dispense, then we do the capping, then we do the liquid dispense, then we incubate, then we can take an image of the vial, then we can also measure Raman and non-contact Raman, we can filter if needed for the sample prep, then we do HPLC sampling, and then we can also prepare in the junior these XRD Kapton plates in case we are doing some salt screening or crystallization. Then we have also developed a solution to close the physical workflow with a mobile robot looking to transfer the plate across the different juniors and the waters, UPLC, and with this integration we will be able to almost double our throughput, because what typically happens is that when we start an experiment, right, we start it early in the morning, then we start the powder dispense, the liquid dispense, the reaction, and typically the reaction ends around 2 to 3 a.m. in the morning, right? So then you need to wait to the next day, go take that plate, just move it to the analysis, and then analyze the full plate. An analysis of the full plate typically takes also a few hours, so then you are basically losing that second day. While if we have the mobile robot, by the time we go back to the lab, the analysis is done, right? So we gain this extra day, right? It's like almost doubling your capacity with a very simple movement. And in our particular case, it's a mirror base with a UR5 arm assembled by Enable Robotics. And here we have a video that you will see the robot in our lab. So this is the isolator where we have the 2 juniors, the powder junior in the left, the liquid junior on the right. So here you can see the mobile robot opening the isolator. Door is a normal door, so it's opening like a human will open it. Then we have there the HPLC sample with a rail that goes out after the reaction is completed. So it will pick the plate, move it to the base, and then close back the isolator. We have also, on top of this anti-chamber nitrogen and vacuum lines, so we can minimize the air contamination of the chamber during operation, but it also is not very, we have seen that it doesn't affect much our processes, because by the time the samples are analyzed, right, and you need to run second generation, it takes a few hours, and in that time it has been regenerated to the specs. So now the mobile robot is driving around the lab. It's looking for our analytical instrument, which is this Waters UPL CMS with the automation portal. It will read the QR code to position itself a bit better, and then it will take the plate, put it in the portal, and then trigger the analysis. After the analysis is done, all that information will go to Catalyst and then to Atinary. We will create a second generation, and then it will run autonomously in the lab. So you will see now. Yeah, that it's taking the plate for analysis, and then the robot simply goes back to the base for charging. So this is the physical automation, right, around the ACDLabs family of tools, the mobile robot, the automation portal of the Waters. Now we need to look a bit more about the other side of the things, right, the digital automation. And as I actually mentioned during the whole webinar, that for us it's crucial, because in order to minimize the bottlenecks, we had to do something, right? Something that was not done before, and we found this solution in the form of combining Catalyst and Atinary in a full integrated platform. This means that now we can generate the ACDLab robotic files that you see here on the left, with the reaction plate, the analysis plates, all the maps, in a few minutes. And you don't need any prior programming knowledge. You don't need to know how to do anything in the Unchained software. You don't need to know any machine learning software to operate the Atinary platform. And Catalyst is also very intuitive. So, anyone with some minimal training should be able to run this workflow, and in order to demonstrate it. So I have here a recording. So it's a real recording of how it takes to run an experiment from scratch using this new integration, right? So we start in Catalyst as we decide, and here we are just adding some information of your experiment. So your department, your site, the purpose, the project code, it's a reaction, and that's it, right? So you can also save your experiment name. So this is just like creating an ELM entry, right? So it's the same. Then you go to your reaction scheme, where Catalyst has a very nice material database. So you can, in this case, for instance, we are trying to mimic that we are doing a coupling, right, reaction. So you can select a bunch of ligands that appear in the database, and then just drag and drop them to the reaction, and then we will do the same thing for all the elements, right? So we are also going to add some solvents for this process. So yeah, butanol, filter butyl ether, etc. So dehydrogenase solvent. So we have them there. Then let's add also some bases to the reaction. So it's just, again, dragging and dropping, simple. Then catalyst. So yeah, we can add palladium acetate and chloride to the mix. So you can see that we have already started to add quite some variables here, right, if we want to optimize all of these conditions. Then for the starting material, yeah, we are just going to do a couple of molecules, and same for the product, right? This is just for demonstration. Okay, so now that we have our reaction scheme, we move to the other side, right, which is the experimental layout. And here is where you define that your reaction plate, and here appears, right, as I said, we do add 24 4 ml well plate. And the only thing that you need to add is your starting material or your limiting reactant to the well, because all the rest of your compounds, right, will be calculated based on that. And now what we do is we select the parameters that we want to vary, right? So we want to vary all the bases as a discrete variables in equivalence from 1 to 3 with a stride of 0.4. And this creates that factor, right, in the DOE. Then we do the same with the catalytic agent when we want equivalence from 0.2. And this is uniform. Then you can do also ligands, right? And they can be continuous, they can be discrete. You can change the units. Typically, we use equivalence. And lastly, I think we have the reactant, right? You can also evaluate, okay, for my co-reactant, I want to study different equivalence from 1 to 2. And we typically also use discrete with a stride of 0.25 and in equivalence. Then last but not least, it's the solvents, right? So we want to also evaluate those solvents. So these are in milliliters and 1 milliliter. So now we have here all of our factor. We'll save it, and here is where the integration starts to kick out, because we are going to send this factor list with all the material classes, with all the information that it's in Catalyst to Atinary by just pressing a widget. So now you can see that the Atinary platform automatically opens it up, and it creates a workstation with already on it all the parameters that we want to vary, that we specified, right? So the base equivalence, the catalytic agent. So we just need to add now the parameter that we want to maximize, in this case, the peak area, and then we create a template, which is very simple. That is, it just guides you step by step. So you will see how quick that is. So you create the new workstation that's been done by the integration. You see here, everything that you want to vary. Then we have we want to maximize the peak area. Next, we can select the optimizer. So this is a very nice thing from Atinary. You can select whatever optimizer you want. Our batch size it's 24. Then we click next, and then we run the campaign. So I think the video is to the next slide. Yeah. So then you just launch the campaign. You can put whatever name you want. You can also preload data. So that's a nice feature from Atinary, that if you have past data, you can use that data, so you shouldn't start from scratch. And here you can see that we have created an optimization reaction with all the conditions that came from the Catalyst integration. And I think it's been 5 minutes, something like that, right? Where we have been able to have all these measurements. So these are all the points suggested by the Atinary algorithm. And now we go back to Catalyst, go to AI optimization, click optimization with Atinary. And what will happen is that Catalyst will read those conditions generated by Atinary, and then you can simply add reactions to container, to the reaction plate one, you select all, and this is beautiful. It's amazing that the well is automatically filled with the composition suggested by Atinary. Before, we had to do this manually, and it took a lot of time for us. And now the final part of the integration is where we are generating the Library Studio file for the Unchained Lab robots with just 2 clicks, right, as you have seen there. So this is to generate the file that the robot will use to start, and it will have the same plate, but in the Unchained Labs format, ready to be run. So this means that anyone with no need of prior knowledge, programming, machine learning, even familiar with the Unchained Labs system, now thanks to the integration, so this is the Unchained Labs file that has been generated, it's able to run these AI-guided workflows, right? Very, very simply. And now, in a few seconds, the file will open. Then we will move to the next section. Yeah, so here you have the Unchained Labs file. with all the maps, all the recipes, and I think I opened it twice. So this is what we will run in our juniors. So you can see how nice this integration works. Then we will have the analysis, right, that will come here at the end of the workflow, and we will send those analytical data back to Atinary to predict a new generation. So all that big area, then you send it to Atinary back, and yeah, there you have it. So this is really an amazing integration, and prior to have this, we have to transform the data into different formats, right? We were always working with Excel files to do transformation. So we add the materials in Catalyst, but then to fill the wells with the data provided by Atinary, so we had to create an Excel file like this, with a lot of manual calculations. Then we also needed to add those to the Unchained labs. So it was one simple set of conditions that needed to go to Catalyst, to Atinary, and to the Unchained equipment, and all of them in different formats. We had to do a lot of manual calculations, so it could take us 4 hours to have everything ready for a simple reaction like the coupling that you are seeing here, right? So, but now it's just a matter of meters. We haven't used an Excel file to do these kind of things since then. No need at all. So I think here we have Sanjay the third question of the poll.
Yes, we do. Thank you very much, Adrian. So Adrian wanted to know, is your organization implementing self-driving labs? Is it something that your organization's interested in? So are you actively pursuing it at the moment, actively pursuing implementation? Is it a short-term goal, something you hope to do in the next year to year and a half? I think Adrian's already shown in what he's discussed that, they went from, did you say you went from no lab and the concept and the idea to now it's like 3 years and you have everything kind of automated. I'm sure you're still working. Yeah, so it's possible. If you haven't started, I think Adrian's showing that it's very possible in the 2 to 3 year time frame. So it will be interesting to know. And I think, Adrian, I think everyone's almost finished, just as I said that, giving us their results. So I'm going to give you about another 3-second countdown. 3, 2, 1, and I'm going to end the poll and we can take a look at the results. So people are actively implementing. Obviously we have a contingent, where this doesn't apply to, but I think that's really interesting. Is that what you're seeing when you're speaking to your peers, Adrian?
Yeah, I think everyone is trying to implement it, even in the short-term goals. They have at least that goal to be actively pursuing this implementation. So we might be in different phases, but I think at least I'm also aware that everyone is looking in the same direction, which is also very nice because it brings more value to what we are trying to do, right? Because this integration is something that anyone can use, right? It's not just for Takeda now. It's something that anyone could use.
Yeah. Well, thank you, and I'm going to pass things back to you.
Yeah, thanks, Angie. So now the last part of the presentation it's just a case study showcasing all of this, right? To show you that it really works and we are actively using it. So the request is simply the example is a very simple backward reaction in the frame of a pipeline LCM project to replace the current reaction, and we didn't screen here a lot of parameters, right? Because we had some background information, but even 6 bases, a few base equivalents, 6 pre-catalyst, a few pre-catalyst equivalents, and 6 solvents. That's over 4,000 combinations. So if you want to do that in a manual way, where you have to screen everything, that will take you quite some time, right? And in our approach, this optimization was done in about a week, in comparison with the 1-2 months of back and forth that will take, with the manual external approach. So we already see here some big wins in time. And looking at the results, this is from Catalyst, the data analysis. So here you can see on the top the heat maps, which it's just the product peak area in different colors, with the color and the scale on the right. So, red a bit of product, orange more, yellow more, and green a lot of products. On the bottom, we have the pie charts, which is the peak area percentage composition for starting materials and products. So obviously, more green, more product. And here you can see that on the left, we have first generation, second generation, and third generation. So you can see that the AI algorithm of Atinary and now our self-driving approach really works, right? So from the very first plate, where we have a kind of a random design, then to the second one and to the third, we start to see more and more green, right? And this is what we typically see. And it's not just that. We also have some very powerful visualization tools. So this one we liked a lot. It's the parallel coordinates graph, and each of the lines represent an experiment that we have done. So one of the conditions, using a base, a pre-catalyst, solvent, base equivalence, pre-catalyst equivalence, and then the results in peak area, and the color represents how much peak area in this case have for your product, right? So you can also filter them, and this very quickly help us to tell, okay, we have potassium acetate and carbonate, and especially BINAP is the only catalyst that works for this reaction. We can use the uramyl alcohol or toluene. We have some freedom in the base equivalence, but we need relatively high catalyst loading. So, all of this information we can get from the data analytics embed into the Atinary platform. And also another one that we like a lot is this map, which it's a 3D, it's a 2D map, a multidimensional map converted into a 2D representation of all the possible combinations that are in the parameter space, being the green points, the one that have been evaluated in the campaign, right? And here, what we would like to see is that this is very important for us for a scale-up, because what we don't want is isolated maximums. For instance, like this point in the corner or this point, right? We want to focus in areas that are here, where you have several points, with good performance, because when you have to do a scale-up and the transfer, you don't want to take the risk of being in this area that you move a bit your conditions and then the yield drops drastically. So this is also a very powerful tool for us. And then lastly, another thing that we do is we tend to do a scale-up of the best predicted reaction conditions by combining the reactol and catalyst to confirm our findings and derive kinetic models. And this step only ends 1 or 2 days of the workflow. So we can, get all the data from the 5 reactors and build the kinetics again in 1 or 2 more days. So we can provide a really good package of data in a bit more than a week. So, summing up everything that we discussed today, right? So with the new emerging technologies, I think, high throughput can be much more than just exploring a few conditions with a factorial DOE and hoping for a positive result hit. But, however, developing this data-rich self-driving lab requires a lot of hardware and software integrations that sometimes are not easy to develop. And to reach this state, we have combined several commercial tools, like Chain, Catalyst, and Atinary, with some internally developed solution to fill the technology gap like mobile road, right? But I think the integration presented here is a great example of how the smart combination of these tools can really accelerate the workflow. And you can see, that especially to bring down the knowledge barrier, right? You don't need to be an expert to run these tools. And while this is not an easy path and has taken us 2 years to reach our current state, we have now the tools in-house, and we can easily carry out this AI-guided closed-loop optimization workflows, being much, much closer to our end state of truly unsupervised autonomous process optimization workflow. I think that's all I have. Thanks everyone for your attention.
Thank you very much, Adrian. I don't know about all you have. That was fantastic. It was so nice to see the actual lab in place too. So we have, I think, we have lots of questions actually coming in from the audience. So I am not going to waste time. We're going to start going through the questions immediately. For anyone who has a question or it's occurring to you, please use the questions panel. That's where I'll be reading them from. So the first question that came in is the, I think it's LSR file automatically transferred from Catalyst to the Junior platform?
Yeah, so we have Catalyst, let's say a shared drive. It's generated into a shared drive that the junior can access. So we don't need to manually drag and drop the files. It's just then you open that file in the junior computer and then you run it.
Perfect. Another question actually from the same person. Why did you decide on the Unchained platform instead of ChemSpeed or Cronect or some other?
That's an easy answer because when I joined Takeda, there were already 2 juniors there. So, and it's also very nice. It's a very nice platform. But we have also 3 ChemSpeeds in our lab, so we are not, let's say, agnostic to any vendor. Like both of them are great and have some advantages and disadvantages. Probably you could do a very similar workflow with ChemSpeed, or let's say in our case, Unchained is our main workforce.
An interesting question here. What was the most unexpected benefit of the self-driving lab?
The most unexpected benefit, probably it was not the self-driving lab as a whole, right? Because we still don't have that, and we still need to do some things manually, right? Like triggering things. We are still developing some integrations, but it was first the amount of data that you can generate, right? There is a moment in time where you keep asking to your colleagues, give me more, give me more, give me more, process reaction to optimize. And they're, no, sorry, wait, we need to pause a bit and process all the data that you are generating, because we cannot, the human cannot keep it up with the amount of data that this machine generates. And the other one was how this simple integration has freed our team a lot of time. It has freed a lot of hours working with Excel, doing manual calculations. I think this is the most valuable things, because now they can focus on more productive tasks, right? So you don't want to have highly educated people, most of them, with PhDs, just running calculations in Excel. It's nuts.
Absolutely. Unfortunately, Excel is something we all love to hate, right? There's a place for it, but usually not in the lab, but it's so easily accessible. That's where everyone seems to go down to. I think, Herman, this might be one for you. They're developing this organization, they're developing their own AI models. What would you say is the benefit of using something like the SDLabs platform instead of their own?
Absolutely. Well, first, great presentation, Adrian and Sanji, Alex. So I want to emphasize that ACDLabs is super flexible to really adjust to what you need, including running your own algorithms on the platform. So we provide our own proprietary algorithms, open source available algorithms, or your own just for your use, so that you can really leverage the user friendliness and precisely, just as you mentioned, why we all use Excel or used to use Excel. Hopefully it's soon in the past, is because it's so easily available or easy to use. Anybody can deploy it. So that's the principle, right? So a user interface that anybody with no knowledge required in AI, in software development, in coding can use it. So we can integrate your algorithms. I would say that's the key, because in the end, the differentiating factors, I don't think are the algorithms per se. They're all perform more or less, depending. Some are better in some applications than others, but that's not really what makes the difference. It's more what Adrian was saying in terms of freeing up time, making it just easy to use. It's in the cloud, so companies that are global, that have different labs in different locations, can all work together at the same time, or asynchronous and have a decentralized lab, if you will. So I hope that answers the question.
Thank you. I hope so too. So another practical question for Adrian: which Raman spectrometer do you use? And what features would you say are important for small molecule SDL?
Sorry, mute. Yeah, it's a very small Raman from Brooks. That it's a non-contact Raman, and the idea that we haven't tested it intensively, but we want to see if we can use this non-contact Raman probe in our for instance, salt screening, crystallization test, to see if we can see form changes, or even when we do solubility, right? We want to provide as much data as possible. So we run a similar workflow, right, trying to optimize a solvent that you could do, right, like with the same workflow, but you're optimizing based on solubility. What we don't want is that at the end of the process, we provide our chemists or engineers, okay, this is the solvent that works. But then they found later down the road that with the solvent, they have a form change. So all they have done doesn't work because the form is not the one that they're looking for, right? So that's why we are trying to use this Raman to see if we can get some information there. So trying to be more data rich rather than, just one analytical HPLC point, to gather as much information as we can to see if we can derive some mechanisms as well. That's why we sample also several times. That's why we take the images to see if we can do some image analysis. That's also why we have the Raman, that it's a bit more complicated to process the analytical data that comes out of it, and we have started to work on it. But yeah, the idea is to not only provide hits, but a very good working package where even the mechanism can be derived out of all this information.
Okay. Do you still see any bottlenecks in hardware or software integration? So I guess with the integrations that you have in place now, do you see any bottlenecks still?
The bottleneck is what happens before and after, at least for us currently, right? To handle the consumables that we expect to do that, but also to do the prep, right? To find all the chemicals that need to be involved in a particular transformation, to put all those chemicals in the deck of the robot, to put the vials in the plate. So that's still a manual work, right? And that's something that we are looking to automate in the next phase, right? The reaction prep part, which now it's probably the most manual time consuming that we have.
Right. And I guess maybe this is something that is related. Someone here is interested in what kind of staff support you've invested in to manage the physical automation of this 24/7 running lab. So I guess people, how many people, like what are the people that you've invested in order to have?
We are a very small team. We are just the 5 of us that I presented, and one is working in Japan trying to build the same thing. So it's only 4 members in Cambridge running all of this, where 2 of them are the robotics engineers doing all the custom integrations. So people running, it's Andrew and myself from time to time. So it doesn't require a lot of people. Yeah, that's the more of it.
Yeah. Your demonstrated workflow only included peak area data for the product. Do you identify and collect data for side products and other things as well?
Yeah, we collect data for everything. So everything is in Catalyst, right? So you have data from your product this was just for the demo, right? But you can add your product, your starting material, your impurity, and the nice thing of Atinary is that you can do a multi-objective optimization. So you can, and we have done it, right? So you can do, I want to maximize the peak area of my product, but I want to minimize this particular impurity to be below this certain level, which is the threshold I must achieve, right, in order to be successful in this reaction. So that it's also something that we can do, adding 2 objectives and using multi-objective optimization.
So I'm going to point out to everyone, we are at the top of the hour. This was how much time we said we were going to take from you. I have so many questions still to go through. Adrian, if you still have another 5 minutes, then maybe we can see if we can get to a few more live here. And then everyone, if we don't get to your questions, then perhaps we'll try and get answers from Adrian, and then I will email them to you afterwards. So we have another question here about databasing. What kind of databasing tech are you using at the back end for storing your contextualized data?
So yeah, the data currently all is in Catalyst, and what we are doing is extracting the data from Catalyst and building our own database. So it's just running a few scripts in Pipeline Pilot, connecting to the Catalyst database, so you are able to do that, and then you can build your own reaction database to do whatever you want, right? Where you would have all the data.
Okay, lovely. We have a question here about scheduling. Are you building your own master scheduler, or are you working with a vendor for that?
We are working with a vendor. we scoped all the options, but it's so broad, especially in the sense that you need to have APIs for everything. We have some of them, but for others you need to develop them from scratch. So we are working for this one for vendors. That's our approach, right? So if we can work with a vendor to do it, and if they have something that works and exists and it's reliable, then we go with the vendor. We do our custom integrations where we feel that there is a gap that we need to invest our time on it.
Yeah. There's a question here going back to, you said, there are things that you're still doing manually. How do you handle reagent storage? Are you using an automated system for that, or does that require manual preparation?
Yeah, so that's the part that I said that's still manual, right? So we have in the big isolator, we have one chamber on the left where we have all of our powders for the unchain in the vials. But still, you need to fill those vials, right, with a new request comes, and also you need to put them into the junior, and you need to put them in the right position, because every time you do a manual step is where you can have an error, right? You might have thought, okay, I put this base here, but no, it's there, right? So that's what we also plan to tackle, and it's our to-do list, right, for this year and next year, try to see if we can automate that pre and post part, right, of the workflow that typically most of these robotics companies, like SMPD and saying, you name it, they typically only focus on the execution of the reaction, not what happened before and after.
Another one here, and this is actually going to be my last question. Folks, I know you have lots of things you want to discuss with Adrian. I'm going to encourage you to use the survey that you will see at the end when you leave the webinar. If we don't get to your questions, we will try to put them to Adrian, but also you can request for particular, to be introduced to Adrian or Herman or Alex. So please use that survey at the end to let us know that you're interested and we can make the introduction. I'm going to take this as our last question. Adrian, how quickly can a new user get proficient with working across your workflow? So using Atinary and Catalyst and the robots, and how scalable is it if somebody really wanted to, grow?
I can answer that very easily, because I said that we are replicating this lab in Japan, right? So Utsan that we hired had no prior experience. He's not an organic chemist. He comes from the biology world. He had never used an unchained or ran a coupling reaction in organic chemistry, right? But thanks that now everything is integrated, we were able to deploy a couple of juniors there. Catalyst and Atinary was integrated, and he's running all of this on its own by himself, with little to none support from our side, right? And it's on another country, you have language barriers, you have time difference, and the training that we had to provide for him was minimal. It was 2 weeks here in Cambridge, and then some minimal training when he was in Japan. So, that gives you an idea of how easy it is after you have done the investment, and if you have done it properly, how easy then is to escalate it.
Yep. You're creating super chemists. Herman and Alex, do you have anything to add? Sorry, the questions were dominated by Adrian.
Wonderful,
Nope.
wonderful work.
Anything to add?
Congratulations. It's a pleasure working with you, with all of you.
Nice.
So thank you very much, everyone, for joining us. I know we went over, but there are so many questions that were still left unanswered. So please use the survey at the end. Adrian, Herman, Alex, I can't thank you enough for such an informative session. You obviously got everyone's juices flowing. So maybe we'll need to do another session in the future and get all of these questions answered. Thank you very much. And thank you, everyone, for joining us. Have a wonderful rest of your day. Bye for now.
Thank you.